thank you all for coming, and thanks for Dean Locke for giving me the opportunity to speak to you guys today. All right, so two things before I get going. First, I'm going to talk about a pretty broad range of sciences. So again, some biology, some geology, some math, some statistics. So if I go fast and you miss something, feel free to raise your hand and ask a clarifying question. No problem there. The second thing is hopefully you will notice that this is a huge amount of work. And so as Dean Locke already mentioned, it's a group effort. So again, this project started about 10 years ago when I sat down at a conference before I even got to West Georgia and had beers with Phil Donahue and Kevin Peterson, and we hashed up this idea for a big project, and then several other scientists joined us on the way. So it's been a huge effort of work. Now, kind of to take a step back and think about where this research started, when I was a kid, I was obsessed with dinosaurs. And I'm sure people know those types of dinosaur children who at 3 or 4 are obsessed with it and can name hundreds of different dinosaurs, and I see them in my classes all the time, and I can pick them out really quickly. Oh, okay, I know that person. That was me. And as I got older and older and stayed obsessed with dinosaurs, I started thinking about, like, what is it about dinosaurs? What is it about these organisms that really, like, enthralled me or picked up my attention completely, which is the fact that they look so strange. Again, if you see a reconstruction of an animal like this is Parasaurolophus, it looks almost absurd, right? It has this giant crest in the back, and it's such a bizarre animal. If you look at reconstructions of dinosaurs, they just look like they're alien, but also this beautiful thing where they're alien in appearance, but also they're real, right? So it's like science fiction, but also something that you could go out and see in real life that used to walk on the very ground that we do. Now, when I started to learn more about geology and taking geology classes and paleontology classes, my opinion on dinosaurs started to shift a little bit, which is not, these are such weird animals that are like unfathomable to almost, well, these animals make sense. They're not weird at all. If you think about it, they're large animals with bones inside their body that pretty much have the exact same bones that we do, right? So, not that weird. You're like, oh, there's this bone in the skull, we have that bone too. Not that weird at all. But as we learn more about Earth history, the further and further we go back, we can still get to that same wonder. Here's Hallucigenia. Much older organism. This is an animal from the Burgess Shale from 500 million years ago at the Cambrian explosion. I'll talk about what that means more here in a second. And this animal, incredibly weird, all right? It's got these tubey things with spikes on one side and spikes on the other side. There's debates for a number of years of which side is the front and which side is the back, which side is the top and which side is the bottom. Again, we have these animals that, for all intents and purposes, look like something you'd see in a hallucination, which is its namesake. And so this looks incredibly alien, which, again, asks the question, like, well, what's going on here? Can we understand these body forms in time? How do we get us and dinosaurs and that weird science fiction thing, right? And so if we think about the history of life on Earth and put this in perspective, life has been on Earth for around 3.5, 3.6, 4 billion years, somewhere in there. Right? The science of when life started is really tricky, but we can pin it down to somewhere around 3.5 to 4 billion years ago. In the vast majority of time that Earth has been around, that life has been around, this is what it looked like. Not surprising, they're fossil bacteria. Right? So in this case, we have this chert. You have little bacteria that bore into this like gelatinous stuff, and then they get crystallized and held inside this mineral itself. Like beautifully preserved individual little cells in these chains. And this is what life looked like for 3 billion years of Earth's history. Right? So again, life has been around 3.5 billion years. Microbes dominated 3 billion. And then we have this event. Right? This is something that's been recognized in geology for a long time, where we go from these types of fossils, which if you looked at the rock like in a hand sample, you wouldn't even be able to tell that there's fossils there. And then we hit what's called the Cambrian explosion, where we go from microbes, imagine going back in a time machine way back to 850 million years ago, you step on and you're like, there's nothing here, it's just bacteria. 510 million years, looking at the Cambrian, all of a sudden, there's an explosion of life. Right? You can see in this picture, again, there's hallucigenia, there, an older reconstruction. There's large predators like Anoblocaris here. To give you a scale, it's about 1 meter long. There's things crawling in the dirt. There's things eating each other. They're competing. They're moving. It's dynamic. And it's a huge shift in terms of what organisms look like. We go from bacteria, bacteria, bacteria to this amazing type of fauna. So let's think about what this explosion is. So it's not so much that this is the first appearance of life, right? Because life goes back further. Really what it is the first appearance of visible fossils, right? Animals figured out how to do something really important at that interval, which is make a skeleton. And once they figure out how to make a skeleton, they can do lots of things, but also from a paleontologic, biologic, geologic perspective, they become fossils really readily. And so if we think about the geologic time scale, this is our first major break in terms of how we break time up. We have what's called the Precambrian. This is the time frame before the Cambrian explosion. Then we have the Phanerozoic, which is the time, Phan means across, Zoic means life. So we pretty much break up the time scale before and after the Cambrian explosion. It's that big of a geologic event. And this is something that's puzzled scientists for a long time, to understand this particular event. You know, Darwin wrote a lot about the Cambrian explosion. He said it's indisputable that before the lowest Cambrian strata was deposited long periods of time, as long as perhaps far longer than the whole interval from the Cambrian age to the present day. Again, he recognized that, hey, we have this huge appearance of all these different forms. There needs to be a lot of time underneath that. And again, he was writing that before we figured out how old things were, the ability to date rocks. And it's still really puzzling. Right? There's almost 1,000 papers published last year discussing the Cambrian explosion. Again, this is an event that's one of the most important events in terms of Earth history altogether. So this is something we really kind of want to get a handle on what's going on. And why is it so enthralling? Well, one, again, we have all these different forms, but the fossils are beautiful. We get a lot of these things called Lagerstätten. It's a wonderful word, right? Lagerstätten. It's a German word that simply means a place to put things. Places where we get beautiful fossil preservation. So one of the most iconic Cambrian explosion types of areas is the Burgess Shale. This is a World Heritage Site. You can't go there and collect, right? It's really closely monitored up in Canada, and we see lots of really interesting animals. You have things like a Shea here, this weird worm that I always kind of envision like the caterpillar in Alice in Wonderland, or Opavinia, this little organism here that has flax along the side of its body, has 5 eyes on top of its head, and this weird little proboscis with little digits that looks like it's always waving. Or we have things like Macaea here. This is an incredibly interesting animal because you can see a head region here, tail region over there, there's a cord running across its back, and these little chevron-shaped muscles. And you can even see these little slits right along there, right along the head. Those are things that define our own fauna. So again, our own relatives are appearing in this Burgess Shale as well. And then we also get things like Morella here, this trilobite-looking organism. And I always love the fossils of Morella because you can see there's the head and all the little appendages. And often we see this big black splotch coming out there. Basically what's happening is if you take this bug, you layer a bunch of mud on top of it, compact it, what happens? Squish. Then we get the little preserved bug squish there within the site. Here's another example of one of these beautiful Cambrian fauna. This one here was just described about a week and a half ago, made a bunch of news reports, and we see this beautiful, well-preserved type of fauna, where we get fossilized jellyfish. There you can see the little cap and all the tentacles, with these arthropods with all these long, skinny little antennae, or here's a Chinorhynchidium, this little worm-like taxa, and here you can see this alien-like reconstruction of what it would look like. Again, thinking about what this looks like compared to the modern day, it just looks patently alien, looks strange. All right, so if we look at biodiversity through time, this is a chart that's just showing how many genera are there as we're moving from the Cambrian explosion forward. And again, if you think about the Cambrian explosion as being like one of the iconic, most important events in the history of Earth, you'd think it'd be some time where something is really changing in the graph. But the Cambrian explosion is that. look impressive at all on this graph, does it? It's a tiny event in terms of the number of species that show up. It's really small. So I'm saying the most important event doesn't reflect this appearance of new animals, or this one, 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 or this one. So why is it so important if we really don't get that much of a biodiversity change? Well, we can change the scale that we're looking at. Here's a paper from 1995 where the question the paper was asking is, well, when do we get phylums appearing in the fossil record? Right? Phylums, think arthropods, or mollusks, or chordates, our own phylum. If we look at where they appear, a lot of them are in the Cambrian explosion, and then there's some afterwards. And a lot of these that have appeared afterwards since the publication of this paper, we pulled back to the Cambrian, and most of these animals here are soft and squishy and not things we necessarily think would get preserved. So what is the Cambrian explosion? It's a time frame where not a lot of organisms show up, but we get a lot of organisms that are very different from them. Again, we get lots of phylums, or first representatives of phylums, and then not that many animals overall. The overall biodiversity is very low. Okay. Which leads us to this term here, which is disparity. Now, what disparity means in a very simple sense, we have diversity and disparity. Diversity, how many organisms, right? So if you look at a study talking about biodiversity, it's talking about how many organisms there are. Disparity is asking how different those organisms are to each other. Okay, that makes sense? So how different in terms of the anatomy and the forms. And so the idea that's been proposed for what the Cambrian explosion is you go through time, you have this explosion of all these different phylums appearing. So right early on, think at this stage here, we don't have a lot of organisms, but they're all really disparate, right? They look different from one another. And over time, we can think about what's going to happen to each of those individual groups of organisms. Some groups of organisms, like the poor Hallucigenia here, is going to go on, and doesn't work out very well for Hallucigenia, and it goes extinct. And that type of morphology is lost forever. Essentially, we have a failed experiment. Life is like, well, maybe spiky and tubey is the way to go. And then it doesn't work. Other groups, like the trilobites, moving forward and then explode in diversity. Because being a trilobite is awesome, apparently early in the Paleozoic. And so now we have tons of them. So if we think about the patterns of disparity that's occurring through time, we have huge amounts of disparity, and then we lose through extinction different groups, while certain groups spread out and diversify within certain forms. So overall, our biodiversity can go up as we increase and have thousands of trilobites, but the range in form goes down because we lose all of these early experiments. And this was a hypothesis brought up by Stephen Jay Gould in this book, A Wonderful Life. Right? So this is the idea of like the pattern of life in general. Okay? So again, we can plot this out, make our hypothesis really clear about what's going on. We have disparity, it's increasing during the Cambrian, and then potentially dropping down, which was the thesis behind Wonderful Life. But ultimately, we don't know. Right? This book was written 30 years ago. These ideas have been in the academic literature for decades, but again, we don't know the pattern. We don't know if the Cambrian is the peak of disparity through time, or if it goes down, or if it stays steady and today's pretty much the same, or if it's increased. We just don't know. Because again, we've never actually tested this idea. Which is a weird thing to think about in science, right? Of like, okay, a hypothesis has been here for 35 some years, since like I was in diapers, but we don't actually have data to directly test this yet. Why? Well, let me give an example why this might be a tricky thing. Here's a crinoid. They're the pinnacle of evolution. They're the most wonderful organisms ever. Because that's the group of animals I love. And you know, how. So, we have crinoids. They have a stalk. They're related to starfish and sea urchins. They have a stalk, they have a body, they have arms. They hang out on the seafloor and filter food. I've been studying them for 20 or so years. I sort of understand them, right? I'm not going to go for, say, I'm the world's expert. No. I'm not even going to say I'm an expert. I'm just going to say I kind of get it, a little bit. Move further away from them, though. Here's an eocrinoid, a Cambrian organism. Okay, it's got a stalk, it's a tiny short little stalk. Has this body that sort of looks like a body, but it's disorganized. Has the mouth there, these feeding appendages that look sort of like the feeding appendages there. So I could look at an eocrinoid and say, okay, I sort of get this. Okay, and it makes sense. And if I had to quantify, put a number on the similarity between a crinoid and an eocrinoid, I could probably figure out a way to do that. But let's move out a little bit further. Here's another Cambrian animal. This is called a Kington. I guarantee no one's heard of these animals before. They lived 500 million years ago and were hanging out largely in what is now Spain. It's related to a starfish, doesn't have five-sided symmetry at all. It's bilaterally symmetrical like us. Has a rim of plates here leading to this little extension out. It's not a tail, it's just a rigid structure. The mouth is probably here, the butt is somewhere, and it has this weird flappy thing in the front. So, how similar is that to a crinoid? I don't know. How similar is that to a crinoid versus an eocrinoid? Can we quantify that? That gets to be a lot more difficult. In a similar vein, going further back, closest related phylum to echinoderms, there's a Hemichordae. It's another worm-like taxa. Has a mouth, has a butt, doesn't have a skeleton. How do we figure out how similar that is to any animals? That's tricky. How do we do that? How do we quantify the differences in form? Again, we could jump to another close phylum. There's me, with a crinoid on my shirt. And we all understand and can easily see human anatomy fairly well, right? We can quantify our cellular structure, our muscles, our tendons, our bones, and compare them to other things like a donkey. I'm pretty similar to a donkey. The bones are all pretty much there, right? There's differences, but we could probably figure out what's similar and what's different between these organisms because ultimately, well, similar shapes, right? Similar morphologies, right? Morphology just means this study of form. How similar are we to stingrays? That's tricky. They have a skeleton, but it's made out of different stuff than you, right? It's cartilaginous. They have fins, they don't have appendages, so that's farther away. And again, all of these organisms are, in the tree of life, pretty closely related, right? So our closest invertebrate relatives are things like that and things like starfish. And then we can ask the question, how similar are any of these things to a snail? That seems like such a difficult question. And again, that's why this hypothesis has stayed a hypothesis for so long, is simply the fact of, how do I mathematically compare any of these things? Right? How do we get a number? Because counting things is way easier than figuring out exactly how different they are. All right. So, what are we going to try and address today? First question, can we use math to analyze these different body plans and make sense of it? And if we can do that, then we can start to map out the patterns of animal evolution over the last 500 million years. And then we can actually address this question of how weird are the weird wonders? How weird is the Cambrian? What the heck is hallucigenia doing in comparison to everything else? And then finally, the million-dollar question is, what is driving this? Why do we look the way we do? Okay? All right. So here's what we did. We used this resource here. Peter Axe is a German anatomist who did a very, very German thing. He wrote a 3-volume set of these thick books in which he systematically went through all of animals and listed out their defining characteristics. Just it was like, I'm going to put all these things in their place, say what makes these organisms an organism. In essence, said, what makes an animal, and listed out those characteristics. What makes an arthropod an arthropod? Listed out a bunch of characteristics. Said, what makes an insect an insect? Listed out a bunch of characteristics. What makes a cockroach? Listed out a bunch of characteristics. They're really amazing books. But ultimately, that's a huge resource then because he's listing out all of these characteristics, and that's data that we can use. So he's systematically listing this for about 396 groups of organisms. Right? So you can see this chart here. You can see he's made a little tree. And you have like Chinorhynchians and nematodes And there's characteristics defining here, or defining these groups, or defining all of these organisms, all the way through there. So he's ultimately, in this book, listing out about 1,700 characteristics of organisms of 212 animals that belong to 34 different phyla. And that's going to be a wonderful resource we can then use to explore animal shape through time. Okay? What types of characters is he looking at? Some of them are cellular, so the shape of an animal's cells. Some are developmental, so how organisms grow. Some of them are skeletal or anatomical features. Right? So there are all these gambits in terms of describing in 1,700 characters what an animal looks like. All the way from the detailed biochemistry to its overall body. So, let's think about this for a second, though. This is a huge amount of information, beautiful amount of information, like you could swim in it, right? Like, you know, Scrooge McDuck doing the back pedal through this amount of information. But also we have 212 animals with 1,700 characters. We have about 374,000 pieces of information. Okay, now we have an opposite problem, right? Which is not, we don't have enough information, but how the heck do we visualize anything? How do we see the pattern within almost 400,000 pieces of information? That's going to be tricky. So we could look at it in every individual character on its own. That doesn't sound fun. Or we could build a multi-dimensional space where every dimension is each individual character. So we have a 1,700 dimensional space. Can you envision that? Can you envision 4 dimensions? No. Like, that doesn't help at all. So what do? Math. We do math because math is the key to everything, right? Okay, stay with me. I will explain it very easily. Okay, so let's talk about the math a little bit. What we want to see is in this big, big data set, the overarching patterns within it. So we're going to use something called non-metric multidimensional scaling. All right, big name there, NMDS. Basically what it's going to do is take characters, right, for 2 particular animals. It's going to then ask how many characters do they share out of the total number of characters. And that's going to establish how similar they are to each other. And so doing that with all of the 200 characters, we can get overall distances between all of those organisms there. Then what this program is going to do, these statistics are going to then try and mimic those distances between things in 2 dimensions. So essentially taking the distances between all those organisms and trying to replicate it through trial and error in a 2-dimensional space that you can see and easily understand. Did that sort of make sense? Yes? Cool. All right, so let's look at a simple example, then we'll look at the complex example. So simple example here. Randomly picked 5 organisms. Starfish, a coral, a clam, well, scallop actually, a bug, and a lingulate brachipod, this little lamb shell. And coded them for 15 characters. Things like, okay, does it have a skeleton? Where is its skeleton? Is it inside its body or outside the body? What is the skeleton made out of? Those types of things. Nice, easy questions that we can ask of these 5 animals. And so I now have a data set of 5 organisms and 15 characters. Okay? We've put it into this non-metric multidimensional scaling, and this is what's produced. Okay, so we get a 2-dimensional plot. The way to read this simply is, if 2 organisms are close together in this plot, that means they share more of those characters. They're similar in terms of those 15 characters that we're defining. If they're far apart, what does that mean?
Less.
Less similar. Perfect. Okay? So in this case, this analysis, based on those 15 characters I picked, says a brachipod's a lot like an insect, and a coral's not like a brachipod at all. Okay? That's what it's telling me in terms of the overall shape. Then you can take those characters and look at how those characters scale to the different axes, and actually get some idea of what these axes might be telling us. So in this case, the organisms down here have an exoskeleton with the echinoderm up here being the organism with an endoskeleton, just like we have. Or organisms over here made out of chitin, like insects are made out of that chitiny material, while the organisms over here are made out of calcite or calcium carbonate. Right? So again, we can start to see what these axes might mean. Okay, so let's scale this up now. Not 5 animals, but 200 animals, and not 15 characters, but almost 1,800 characters. And that's what we get. Right? So a plot there, we can read this very simply as these two organisms close together, similar in terms of those almost 1,800 characters. This organism and this organism, super different. Okay? But wait a minute. There's another thing we should think about, which is this idea of non-applicable data. And the reason I want to point this out real quick is in a data set like this, this issue of non-applicable data took about 2 years to get passed. So let's think about why this is. Say you have this animal here, it's a comb jellyfish. Right? It's a jellyfish, instead of having a bunch of tentacles, it has these little ridges on it that it uses to swim around. Beautiful organism. Right? Say we have a character. Say we have a character that says heterodonty, present or absent. Heterodonty, let me explain what that means, is hetero means different, donty means teeth. So it's asking, do they have teeth that are the same across their jaw? Right? For instance, do you have heterodonty?
Yes.
Yes. Take your tongue right now and do this. Are they different? Yeah. Your front teeth look different than your back teeth. That's all it means. If you have a crocodile, their teeth all look the same. So a crocodile, absent for heterodonty, us, present for heterodonty. Make sense? Easy way to code that. Does a comb jellyfish have heterodonty?
Doesn't have teeth.
Thank you. It doesn't have teeth. That question makes no sense. Right? It's like me asking what color are your feathers? Like, Brad, what are you talking about? It doesn't make sense. So what we need to do is establish that we're not saying that this ctenophore doesn't have heterodonty. We want to say it doesn't have heterodonty because it doesn't have the equipment, the anatomy to even make that question make sense. Okay? So that's nice and makes sense what I'm talking about, but this is what we then had to do. We had to take those 1,700 characters and ask how all of those 1,700 characters relate to the other characters. In essence, make a huge map of, if it has character 1,503, that requires it to have this character, and that character has to require it to have this character, and essentially find out the interrelatedness of all of those characters. Ugh. And so we did this, took this graph here, and ultimately it changed it to that. Basically, if you look between these, much to my chagrin, we did 2 years of work, so largely done by my colleague Phil over in Bristol. We still have the overall general shape. We just take about 4 taxa from this big cluster of things here and move them out. So it's like you spend 2 years and then like 4 data points move. It's very unsettling. But there we go. We still solved this issue. So let's look at this space here. Now, what we have are eukaryotes, so single-celled organisms. We have sponges there. And then we have our jelly-like animals here. So our ctenophores, our jellyfish and things here. Then we split. In this direction, we go towards the mollusks, like clams and snails, to velvet worms, here with the Onchophorians, and then over here with the arthropods, the bugs and crabs, and things like that. Here in this big cluster of organisms right in the middle, they're all pretty much wormy taxa. So we have like 20-some phylum all sitting there, but if you look at them and kind of squint your eyes, they all look essentially like a tube with another tube around it. Right? Front end, back end, gut, body. That's largely what all these taxa are here. Then over on this side, we have the echinoderms, so our starfish and things, leading up to fish and us. Right? So we can actually map out all of these organisms groups. There's some pictures to guide our way through there. Again, sponges, jellyfish, wormy things, mollusks, bugs, crinoids, yay. And then all the way up to birds up there on the top. Okay, so again, we now have our map of all these organisms based off of these 1,700 something characters. Let's zoom in a couple spots. Let's look at the bugs more. So here we can see the bugs, right? The overall arthropods, and we can see the spacing out of these different groups. Down here in the green, we have the chelicerates, which include things like sea spiders beautiful organisms here, and horseshoe crabs, and spiders, and scorpions. Here we have the crustaceans, again, crabs and shrimps, and brine shrimp and all these other things sitting there. We have the myriapods. Myria means a lot, and pods means legs. So we have our centipedes and millipedes up in this area. And then up here we have our hexapod. Hex means 6. There we go. So we have 6 leading up to this giant cluster here, which is insects. Right? So we can see this space and actually say, well, overall, all the insects look a lot like other insects. Shouldn't be a big surprise. And then we have things that are very different from one another. A scorpion and a wasp look quite a bit different. Other side of the tree. Here's our own group. So we can start off here with our closest relatives that don't have backbones, things like the sea squirts here and the lancets, leading up to the jawless fish, so there's a hagfish right there. Big jump over here to the cartilaginous fish, sharks and rays, to the bony fish here, to the lobefin fish like the coelacanth, up to the amphibians, the mammals, the reptiles, and birds all the way. Again, we're mapping out these very different organisms across the space. Okay. So, now let's add the weird stuff. We also looked at about 70 animals from the Cambrian. We basically looked at the entire diversity of what's there through this time frame. All the way from our familiar friends, the Opabinia here with the 5 eyes and the weird proboscis, with Lucigenia, to these worm-like taxa, to Anomalocaris, the mega predator of the ancient sea, to ancient sponges, ancient brachiopods, our own relatives like Metasphragina here, this kind of like almost hoping to be in a few million years a fish, and then things like these weird ancient relatives of starfish as well. So we have this huge diversity of body forms that we have in the Cambrian, and we want to add those into our analysis. The other thing to point out, a lot of these are from those beautifully preserved areas. Again, remember the Burgess Shale and those new deposits in China, we're preserving things like jellyfish, right? So again, we have a lot of information about these organisms. Okay, here's an issue. We have cellular characteristics or developmental characteristics. In essence, we're asking what's the details of cells, which doesn't get preserved, or details of how those animals grow, which often doesn't get preserved either. And so in total, about half of the characteristics that we're talking about aren't things that are going to show up in the fossil record at all. So this creates a problem. We can code them as missing data, just tell the program, I don't know. And what that does, here's the red dots, here are the fossil organisms, is in all of these cases, they get pulled down in this direction. Right? We can see these red dots skewing downwards. Essentially, losing those characteristics makes them look more simple, because we don't have that information to add. Same thing, if we only limit the data set to the 800 or so characters that we can see in the fossil record, these fossil organisms still skew downwards. And this is a pretty common problem in paleontology. If we have evolutionary studies and we're trying to figure out how things are related, the fossil record produces this bias that makes fossil organisms look more primitive than they actually are. So we can see that issue here. We can see this issue lots of places in paleontology where the fossil record is kind of misleading us. So how do we get around this? Well, what we can do is take the tree of life, which is different than the tree proposed by Peter Axe in those books. It's based on modern day studies of our understanding of how things are related, and plug all of our animals into this tree of life, and then do some more statistics and try and model out what those missing characters would be. This sounds a bit iffy, right? That we're going to try and model out using statistics of what those states are going to be. But in a lot of cases, it's just telling us that things are inapplicable that we think should be inapplicable. Right? Like, okay, well, this type of cell is only there in snails. I'm okay with saying that it's not there in a jellyfish. Those are the types of assumptions that we're making in this case. So we're not saying too many features are going to be there or making up morphology. It's mostly just like confirming what we already think. The other thing that we can do with this method is model out what all of these potential ancestors, all of these branching points in the tree would look like. In other words, we can ask, hey, what's the ancestral vertebrate look like? Or what's that, ancestral snail look like, and model out the characters that we think should be there. Then we can plot those out into this multidimensional space. So again, here's our data set of animals. We're going to take this tree and plot it onto that. And that's what it looks like. Let's think about what this is, though, and why this is really interesting. Again, 700 million years ago, we just have single-celled organisms. Right down here in the space, as we move forward, we start to see sponges around 650 million years. Then we start to, around 550, see things like cnidarians, right? So see coral-like organisms. Then we hit here, and we have this major split between organisms. Biology majors, geology majors, that's our deuterostome, protostome split between major groups of organisms. We have this huge explosion of life here, and then we have this movement over in this direction towards our bugs, right? That's happening at some point in Earth's history. And again, we have this steady progression up to us. There's other things we can see as well. We can see this group of worms here, their ancestor was all the way there, and they evolved to look like something that already exists on Earth. Essentially, they converge back on this wormy form. So we can see convergent evolution, we can see the exploration of different body plans through the last 500 million years. Essentially tracing out the path that life took. Here I've added in the fossil organisms, which are in black dots. So there's a dot there, dot, 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 all through there. And we can actually say what some of these dots are. There's Metaspergina, this weird, hopeful fish thing sitting right there. Ophibinia with the 5 eyes sitting there. There's Halicarids there, which is the picture from the first slide. And Morella and trilobites are hanging out over here. So what can we say with this? One, they're not on the outskirts of, the space. Right? We think of Hallucigenia, that weird spiky wormy thing, as something that's like way outside of all of other life forms. It looks alien. It's hanging out here. Right? This animal here, Ophibinia, 5 eyes, proboscis, flappy things. Looks weird. Again, it's central in our space. So let's ask some questions then about what weirdness means. Fossil organisms. Ophibinia looks weird, right? If you were scuba diving or snorkeling and saw that swim by in a weird rippling pattern past you, you'd be like, what the heck? So it is, to our eyes, something that seems weird. And I make the argument that, sure, it's weird, because ultimately, nothing is alive today in this space at all. So it's not that it's an outlier in shape space, it's just that that space is unoccupied because everything that looked like that is dead. Right? So this extinction is the thing that makes these weird animals weird to us, because they're all gone. The other thing that this extinction does, with this death of all these organisms in this area, makes it so it looks like our phylum are more distinctive than they actually are. Right? If we think about the relationship between velvet worms and arthropods, today, our closest thing looks like a millipede, so it doesn't look like them at all. However, you have an arthropod here and a basal velvet worm here. All of a sudden, they're not that distinctive. So again, extinction makes our phylums look more different than they were in the past. Again, extinction makes Ophibinia look weird just simply because they're dead and gone, not that they're objectively different. All right. And here in this graph here, I've just outlined the shape of the Cambrian explosion. We've moved past what the world looked like in the Cambrian, which kind of, if you think back to that initial idea, makes sense, because evolution didn't stop 500 million years ago. Big surprise, right? Evolution continues on through time, and there's multiple instances of things sneaking out there, sneaking out there, sneaking out there, past this Cambrian envelope. So again, the unfamiliar is weird, but the unfamiliar isn't actually weird. If that makes sense at all. Still with me?
Yeah.
Yes, cool. All right. Why do animals look the way they do? All right, now that we have a good handle on this, let's now talk about why we see these features. Why did this occur? And there's two ways to look at this. We could look at the things that are extrinsic to these organisms that could be driving change. Things like oxygen levels. If there's more oxygen, they can get more food and get to bigger sizes. Nutrient levels, we see this influx of nutrients around the Cambrian explosion. We could see ecology, so predation and escalation, this arms race between predators and prey. Alternatively, we can look at intrinsic things, like the genetic material, the genomes of organisms, or regulatory genes for proteins, and ask what is causing and driving this change. We're going to focus here on the second bit. Okay, so we're going to shift into some major biology here. Okay. Other thing to mention, we're going to be looking at comparisons by looking at the origin of the different phylum. So again, we're equalizing things down. So this is what our morphospace looks like. We just plot out the 34 phylum instead of all 200. So again, similar type of graph, just plotted down. Okay. Let's talk about some statistics, which for my colleagues and students, maybe you already know this, but general things that are really important to talk about. Because I'm going to talk some statistics, again, back to math, but let's talk through a real quick primer. First thing, these events that we're talking about happened over the last 500 million years, therefore, they're beyond our view of what we can test in the lab, right? We can't test this by making a model ecosystem or world and then like watching it for 500 million years. Can't do that. So again, we are essentially asking about the related patterns that we can observe. That means we can't prove a darn thing. All we can do is ask, does the data support our ideas? And ask, essentially, if there's a random data set that has some normal distribution, is the relationship between our ideas and the data more extreme than just randomness? Right? That's what we're doing here. We're asking, is the data more closely related than essentially 95% of random data? Right? So that's our idea here. This idea of a p-value, which you might hear about, it's basically asking, is my observed value more related or more on the extreme than 95% of other randomized data? Okay, that's going to be our metric. A couple of things to keep in mind. If you increase the sample size, then it becomes easier to hit that 95% cutoff. So if you have a huge data set, you can find a significant relationship between something that's really not there, just by collecting a huge amount of data. So you also have to see the effect size. So how much of a difference are we talking about? Multiple tests. Let's talk about this real quick, because I guarantee you've seen examples of scientists misusing this. Let's say we have a 1 in 20 chance of attributing a correlation to something based on randomness. Again, that's what that probability means, right? 1 in 20 chance of relationships. Say we then do 20 of those tests and compare it against 20 things. What's the chance of getting a relationship and interpreting random data? Well, if it's 1 out of 20 and we do it 20 times, our chance of getting one of those is 1. So for instance, if I take a study, take a whole bunch of people, and ask how much coffee they drink, and then look at their blood pressure, look at their BMI, look at a whole different number of these health statistics, what's the chance that we'll find one of those statistics that's correlated? Pretty darn good. That's why you keep seeing these studies that's like coffee's good, coffee's bad, coffee's good, coffee's bad. They're doing the same problem, right? They're just throwing the statistics up against the wall and then figuring it out, and finding some relationship that's pretty much random. All right, correlation and causation. This one you guys are familiar with, right? Just because two things are correlated and we see the same pattern through time does not mean one leads to the other. And ultimately, that's how science is done, where we're not looking for that correlation because then we can prove that those two things are related. Instead, we're looking for the inverse, which is if two things are uncorrelated, then you can be pretty certain and say that there are probably one isn't causing the other. So we're looking to falsify things, prove things wrong, rather than say the pattern exists. Does that idea make sense? So we're trying to not do that. Okay, so the test that we're going to use is called the Mantel test. It's looking at distances between things in multidimensional space, and looking at the distances and asking, do we have the same distribution? So in other words, it doesn't matter where the organisms are, left or right. It's asking how far things are apart. Are similar organisms close together, or are they far apart? Okay? Okay. First thing, let's talk about a null hypothesis. There's this idea that came up a couple of years ago that time plus random evolution equals more disparity. So our base level here is you don't need anything really, just more time. And what we found is that the amount of disparity within a phylum is unrelated to when the phylum shows up. So again, you have sponges here that showed up 700 million years ago. Well, they hardly generate that much shape change. So again, something more has some common. What else? Genome size. How big is a genome? And the surprising thing about genome size is that the length of your genome, the amount of DNA you have, is actually unrelated to how many genes you have and you use. Surprising, right? You don't use most of your genes. They're just sitting there. Never coded, never expressed, they're just there. But there is an argument that these non-used parts of your DNA are how you make new genes, right? So through mutations of these things that you don't use, you can make a new gene that does something brand new and novel. And what we found is by comparing 22 of the phylum where we had data on genome size and data on their morphology, that we had a probability that these things are closely related. The distances match up pretty well. So new genes might mean that we have correlation between what genes are important, what proteins it can make, or new genes expressions, how they're regulated. And real quick, pretty flower there. Longest genome that we've ever discovered. It's like 5 times longer than yours. Why? Definitely does not use most of those genes. Okay. Proteins. So proteins are the ways that we express features, right? As you're growing, as you're a tiny little embryo, you have genes that make proteins. And so we can make new proteins through time, and the idea then is, well, maybe certain groups of organisms figured out how to make more proteins, and that made them different. Okay? And so my colleague Jen Greenwood, as part of her PhD, compiled a huge list of proteins, the superfamilies, diversity of proteins in these different groups and families, and the different shapes of the proteins and which ones they had it, and found that none of them were correlated with the shapes of the organisms. So if you have lots of different proteins, hey, it doesn't matter. If you have certain shapes of proteins, doesn't matter in terms of what the organism actually looks like. They're not correlated, so probably this is not driving our trend. All right, third thing. So if it's not proteins or the genes we have, it might be how those genes talk to each other. Okay, so let's talk about this. Here you have an embryo, right? This is what you look like at a couple weeks in. Adorable, right? You have a head, you have a tail, you have some limbs. Good, you're a growing little baby, okay? Now let's think about how this process works, of how you grow a person. So as you're growing, let's focus in on this area right here. So as you're growing, you form a little bud, it's a little limb bud, and then as you're growing, there's a gene right here that's expressed in this part of your hand. Just so happens the scientists who discovered the gene called it sonic hedgehog. Okay? I'll talk about why that is for a second. So sonic hedgehog is activated, and it sends out a signal, right? Essentially, it sends out a signal, that's our transcription factors, that's a single signal going out, and that signal is going to activate other genes. When it's close to the signal, there's a bunch of the signal there, it's really loud, they activate one set of genes. When it's far away, it activates a different set of genes. So, where it's close, it starts making a pinky. Then over here, causes cell death and causes a gap between fingers, and over here it makes a thumb. Right? So that's a really important gene then, this operating gene, this regulatory gene there. That makes sense? It's cool, right? And we can understand birth defects. If you turn up that gene, you can make 6 fingers or 7 fingers. If you turn down that gene, you can make a pattern. Be real mad science about things, right? One thing that we can point out though, the ways these cells communicate, those transcription factors, are found in single-celled animals and pretty much shared across all life. Those big genes that are organizing front, back, left, right, making eyes, making appendages, those are shared across all organisms, all like animals. So again, those can't be the things driving this change. Right? Ultimately, we found sonic hedgehog based off of a gene in a fly.
called hedgehog that makes the front end versus the back end, one side slightly hairier than the other. Right? It's the same general operating principle. Okay. Where do we see differences? We see differences in these things called microRNAs. So let's think about how these work. I'm going to use a metaphor here, so stay with me. Say your body is like a car, right? And that's made at a factory. So an individual gene has a function, it makes a protein. It's like the person at the car factory that takes a muffler, welds it onto a car. Cool? Then there's someone who tells that person how to go to work and when to work, right? It's like the boss, the foreman at the factory. That's like Sonic Hedgehog. That person says something, it's a transcriptome factor, it's communicating, getting that other gene to work, welding it on. Okay. You could take that person that's the foreman, move it from a Ford plant to a Chevy plant. That new plant doesn't start making Chevys, right? It is essentially moving across there. We can transpose them across different types of organisms. Okay, who else is at the factory? Chances are there's someone up in an office that's looking at sales figures. Going, oh man, these cars aren't selling as fast, we better slow down production. Or man, this has been a great month, let's turn up production. And so they send a message to the foreman that tells the foreman how much the people should work, that then trickles down. That's what these microRNAs are. They down-regulate regulatory genes. In other words, they tell the regulatory genes how much to work. Okay? Let's look at an example in the real world. Here's some fly bites. Cool, right? Back end of a fly. Normal flies here have 1, 2, 3, 4 hairs coming out. Okay? Out in nature, that's what we see. If we disable the microRNA, so that microRNA that's operating in this part of the body doesn't work, they vary wildly. Some of them have 4 hairs, some of them have 5 hairs, some of them have 6 hairs, some of them have 7 hairs. So we have this huge variability. If we turn up and increase the amount of microRNA operating there, you get a single hair. So what these genes do is they focus in on general shape and always hitting on the exact ideal shape for that adaptation. Cool? That kind of makes sense? Get a nod? Yes?
Yeah.
Cool, still with me? Good. All right. These types of microRNAs vary wildly across space, and if we code them all out and analyze them in the exact same way, we get this shape here. We get wormy taxa in the middle, one big thing out here for the vertebrates, over there with the simple animals like the sponges, and over in this area to the snails, mollusks, and bugs. That pattern makes sense? That's the same pattern as the morphology. And when we correlate them together, we get this really nice p-value, showing that these organisms, again, are doing something very different. Things like vertebrates and arthropods have a ton of them. Mammals have a ton of them, right? Which allow us to understand this. That maybe it isn't so much developing new genes, but figuring out how genes are communicating to each other. All right, so causation. Again, there's probably other things that might be in this as well. I'm not saying gene regulation is the only thing, but this does allow some further testing. Okay. Other mechanisms. If we look here, here's our Cambrian explosion, and if we want to look at the animals outside of it, here, 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 and here, one thing that they have in common, can see how they're more red than blue? The red ones are the ones that make it up on the land. So the other thing we can say is, okay, the Cambrian explosion, we have these different things going on, making up to land is really important. With bugs making up in the Silurian, and then our own group like Tiktaalik here, making up to land afterwards. So to conclude the talk, this is what I want to do. I want to reframe the entire question. So, at the beginning, I showed animals like this. Here's Microdictylon. From the Cambrian, weird, right? It's a tubey organism with appendages. These things have been interpreted as compound eyes. Whoa. How ordinary. It's in the middle of space. It's a tubey organism with eyes where it shouldn't be. Yeah, in nature we got lots of those. Right? Arthropods do that all the time. This animal, not interesting at all, right? Sitting there in the middle of space, eh. You know what's weird? Butterflies. Man, they're crazy if you think about it. They have this tiny little body, long legs, huge wings. They're up on land, that's already weird. Then they have like a crazy straw that they fly around to drink pollen and nectar. Weird. You know what else is weird? You. You're a huge vertebrate living up on land with a skeleton on the inside. So strange. This giant big brain. Yeah, we're weird. We should reframe this whole question. These Cambrian things, nah, they're not that weird. We just don't see them anymore. They're long dead, which they lived 500 million years ago. Big surprise they're dead. We're weird. We're sitting on the outskirts of that morphospace. So, why are we weird? Two things. First, way back in our history, 500 million years ago, our ancient ancestor figured out a way for the genes to talk to each other. That gene regulation, those microRNAs, had a new way that enabled these organisms to figure out how to make new features. Right? Figured out and basically how those genes communicate and do things that are different and allow them to evolve into new forms. What else happened? We made it up on the land. Our ancestors, not 500 million years ago, but 350 million years ago, ventured up on land. Like Tiktaalik did the first little floppy things to pull himself up on the land when it looked around and said, man, there's a lot of big fish out in the ocean and there's a lot of bugs not being eaten up on land, and it kind of flopped forward, right? And that's an incredibly important thing because then there's new niches up on land, new ways of making a living that there's no competition for. And so they moved up in those spaces. And the other thing that's really fascinating is this also shows us one of our iconic things increasing, which is what makes us us, which is intelligence. Why is this? There's this cool paper that came out a couple of years ago that looked at the size of the eyes of some of these low-fin fish to amphibians as they transition onto land. And what they found is the size of the eyes increased a huge amount when they made it up on the land. Why is that? If you're in the water and you start swimming around without goggles and you open your eyes, how far away from you can you see? Not far at all, right? So if you're a fish and swimming around, you see something, you have to react instantly because it's right there, and your choices are essentially, it's small, I'm going to bite it, or it's big, I better swim away. Right? That's your choices. And then you go on the land, and your vision increases by hundreds of times. Now you can see 10 feet away. And so you look, and you can see all the way on the other side of the room, man, there's a tasty, delicious bug. Now I have choices to make. I need the intelligence to think, should I stand here and wait for that bug to come to me? Or should I go to the bug? If I go to the bug, what's that bug going to do? If I go this way and it veers off that way, so I actually have to think about the future. For the first time ever, like thinking about that concept of, what do I do? And that choice leads to bigger brains. And then bigger brains, and bigger brains, which leads to you smarting. So, with that, I want to stop today. Thank you for your attendance. Thank you for your attention. If you have questions, I'd love to answer them. Okay. Questions? Yeah.
In the NMDA graphs, how many traits did you have on each axis, and how did you decide which axis to group the traits on?
Oh, so this, basically in that case, it's looking at mimicking it all entirely. So it's not even placing the traits on the axes. It's just saying, okay, I have this axis, and I'm trying to mimic the overall distance structure. So in that case, one of those axes might represent, 700 different characters. But again, that's not quite how it does it. It's just saying, okay, I know this animal's this similar to this animal, and this animal's this similar to this animal. I'm going to try and fit them on the graph to keep those distances fairly consistent. Straight off the bat with a math question. I love it. Go for it.
Measuring intelligence seems interesting to me, and I guess I can only relate to the more common animals that we know today. But are you saying that one of the ways that scientists have measured intelligence is based upon the size of the brain relative to the size of the animal?
To a certain extent.
And
Yeah.
you think about them having so many more other senses that we don't have. So how do you really measure intelligence, you know?
Yeah, that's a tricky question. So measuring intelligence out in nature, super hard. So we can do it really well with animals that look very much like us, right? And have senses and abilities very much like us. Like we can compare our intelligence pretty well to a chimp's intelligence or a dog's intelligence. Where it gets really tricky is when we leave our own phylum to animals that are very different. Like one of the major tests for intelligence in animals is you take an animal, they look at a mirror, you take some paint and put a dot on them. If they like try and rub it off, you're like, okay, they realize they're looking at themselves. Which chimps can do, but dogs can't. If you have an organism that is completely solitary, but super smart, but solitary, and also communicates by changing the color of its body, that test is meaningless. In other words, if you have an octopus, and it sees a dot on itself, it's like, man, that's pretty common, I do that all the time. Right? So again, how do we figure out how smart an octopus is? Yeah, it's hard. And again, like there's a lot of things that correlate along with intelligence. Like their ecological position, because hunting your food takes more smarts than like a cow finding grass. Right? Or the more societal interactions there are, or the more complicated environment that they live in, or the more complicated behaviors that they have, again, correlate with intelligence. But that's a good question, because that's a really hard thing to actually scientifically get at. Yeah?
So from your research, can you see into the future? Meaning, can you see what we're going to look like in a million years?
Oh! I mean, no. The cool thing about this is if you look at those evolutionary trajectories, they don't have to go in any particular direction, right? They can expand outwards or they could go backwards. So they could converge on a point from very different ancestors and converge together. So where do they go from here? Again, no idea. If you want my opinion about human evolution, where we're headed, that's a tough question, right? On one hand, you could say easily, evolutionary change happens when you have small population sizes, and we have like 7 billion, right? Also, we see change when there's strong selective factors, and we, figured out how to bypass bad eyesight. So maybe that causes slower evolutionary rates in us. And ultimately, are we going to keep evolving or go extinct? That's a super good question, and ties into this as well, because ultimately, all those animals, all along that trajectory, you can now imagine if we went through from the Cambrian, Ordovician to Silurian, Devonian, all the way through, it's a lot of dead things on that graph. So, are we going to be one of those dead ends or not? I don't know. Need a time machine. Yeah.
So, just thinking about our place in this universe, when humans, before Copernicus, thought that the sun was the center, or that the Earth was the center,
Yeah.
and planets revolved around us. So this sort of is the same thing to me, because suddenly we're the weird ones.
Yeah. So,
Do you think that's going to cause a shift in the way humans think about ourselves, instead of we are the princes of the universe?
So we can think of this in two ways. One, we're definitely on the outskirts, right? And so we're up in here, not in the middle. We're not the center and gravitating outward. We're on the edges. Cool, right? We're like one of the most bizarrely evolved things in the animal kingdom. So that's one thing there, putting us out to the side, even if birds are even further than we are. Okay. The other thing to mention that I really like about this, and we'll finish with this today, is the fact that we're connected to everything, right? Ultimately, if you look at this spreading of data across here, everything is connected. We're closely connected to our close relatives, to things like other mammals and reptiles and birds and fish, right? There's us to fish, but also to worms and to starfish and to bugs and to insects and to sponges and to singular stuff and things. So not only is this showing us where we are, but it's also showing us in relationship to all of our distant relatives. Again, everything is connected. And again, this is just our extended family tree. So again, everything's connected, kumbaya, we're all there together. Cool. So thank you guys very much, and appreciate your attention.